So I'm Sara Tolaney. I'm a breast medical oncologist at Dana-Farber Cancer Institute. And today we're going to be talking about TROP2-directed antibody-drug conjugates and their role in treatment for metastatic breast cancer, both in ER-positive as well as in the triple-negative setting. So we'll start off with a case. So this is a 55-year-old woman, a patient of mine who originally had presented with an ER-positive, node-positive breast cancer. She had gotten chemotherapy and at the time had then enrolled on the PALLAS trial. That was the trial that involved giving endocrine therapy with or without palbociclib. And she had gotten randomized to get it with the palbociclib. And so she had been on that, but after being on treatment with endocrine therapy for a little over 3 years, developed elevated liver enzymes and was found to have liver metastases. So, biopsy of her liver did reveal ER-positive disease that was HER2 1+. We had done an genomic testing, and she did have a high tumor mutation burden that was over 10. She did not have any evidence of a PI3 kinase mutation or ESR1 mutation. And so, given this was now first-line treatment for metastatic ER-positive disease, we started fulvestrant with abemaciclib. But she didn't have any benefit to this. And in fact, her liver enzymes were elevated when I started therapy, and they actually continued to rise over the weeks after starting. And so, of course, I got quite anxious and reimaged her, and she was not benefiting from therapy. And so we started capecitabine. And she did have improvement in her liver function then, and things started to turn around. And in fact, she had a great response to capecitabine and was on it for a little over a year, but then developed disease progression and had worsening disease in her liver and bones and was actually symptomatic from the bone progression. And so we actually had quite a debate at this time. This is a very educated woman. She actually is a comedian by training, and so she's also quite funny. But we had this debate that she was HER2 low and had now had one line of chemo, and so she is a candidate for TDXD or trastuzumab deruxtecan, and so that was one choice. The other choice was with the time we had a clinical trial called the SASI-IO study, which randomized patients to get sacituzumab with or without pembrolizumab. And given that she had a high tumor mutation burden, she was very interested in trying to get combination therapy that involved immunotherapy with a chemotherapy like agent. And so she really wanted to go on the trial. And so we decided to put her on the study. She was quite upset, though, that she got randomized to get the sacituzumab alone. So she didn't end up getting the arm that she wanted, which was to get the immunotherapy. But it turns out she had a great response to the sacituzumab, and in fact had what we would call a partial response by RECIST by the trial standards, and actually was on study and just recently actually progressed 2 weeks ago. So she had started to have some slight progression within her liver. It was actually just one area in her liver that had progressed. Everything else was doing okay. And we did actually biopsy that area in the liver that had grown, and it still was ER positive and HER2 low. And so, really we're now faced with the decision of what to do next. But I thought it would be helpful to think a little bit about the TROP2 ADC that she got and sort of how we think about utilization of these agents in hormone receptor positive breast cancer.
Just before you go on, I was just kind of curious. You said the TMB was high. How high was it? And what was the PD-1 assay?
So good question. So her TMB, I believe, was 11 or 12. We didn't actually test for PD-L1 using a commercial assay. The trial did test all patients for PD-L1, but it was anonymized, so I don't have the result of her PD-L1 assay, so I don't know that status. But yeah, it definitely did have a high TMB.
I mean, I don't know exactly when this happened, but if this were to happen right now, I mean, would it be consideration just to give her an IO based on the TMB initially, instead of going on the trial?
So she actually progressed 2 weeks ago, and last week she did start immunotherapy. In fact, we had this other debate, which is, pembrolizumab has a monotherapy indication, right, for high TMB, but tumor agnostic approval. So you just give pembro alone. But my worry is that the response rate for pembro alone was only about 20%. And we do have some data for combination therapy in breast cancer where it seems like when you give chemo with checkpoint inhibition in general in breast cancer, we seem to do better than just checkpoint alone. So I was actually really interested in trying to get her combination treatment, but there's no FDA approval to do that. So I will be honest, I was a little creative. We had done a trial a way back when with eribulin plus or minus pembrolizumab in hormone receptor positive disease. And in fact, when you looked at the subgroup analysis, it was the high TMB patients that seemed to have benefit to the treatment. So I argued with her insurance company that there is actually a little bit of data here, and they actually did approve. So she did just start on eribulin plus pembrolizumab in this setting.
Wow. What a story. Okay, so let's get into the stuff, I guess.
Yeah, so I think we were really, again, while this patient was not happy to get randomized to the sacituzumab by itself, I think we were very fortunate that she did have a great response. And so, in general, the way the sacituzumab is working is it's an antibody-drug conjugate that's targeting TROP2, and it has an SN38 payload, so a topoisomerase 1 payload. So in fact, it's the active metabolite from irinotecan, a drug we're all very familiar with. And it has a very high drug-to-antibody ratio of about 8-1. So it's internalized into the cancer cell, you get payload release in the cancer cell, and in fact, that payload can diffuse through the cancer cell membrane into neighboring cells, what we call the bystander effect. And so, again, is able to deliver a lot of chemo into the cancer cell. And we know that the vast majority of breast cancers, in fact, have TROP2 expression. So if you look at triple negative breast cancers, it's 96% of patients have TROP2 expression, and hormone receptor positive disease, it's 95%. So almost every single one of these cancers has some level of TROP2 expression. So it becomes a really attractive target because this is expressed on cancer cells and not so much on normal cells. And it turns out there are now three main TROP2-directed ADCs that are pretty far along with their development. Sacituzumab obviously has approvals both in hormone receptor positive and triple negative disease. Datopotamab deruxtecan has positive data in hormone receptor positive disease, and we're awaiting approval. And sacituzumab tirumotecan, now called SAC-TMT, is now moving into phase 3 trials, is another TROP2 ADC. So nice to see so many of these agents.
Yeah, I was struck by that third one. Like, what's the difference between that and sacituzumab?
Yeah, so it's interesting. It's also targeting TROP2, and the payload is a belotecan derivative. So, another topoisomerase 1 inhibitor, not the same as SN38 and not the same as deruxtecan, so slightly different, but also a topo 1 payload. And the drug to antibody ratio is almost identical to sacituzumab. So, they are very similar in construct, but, as we'll talk about, the data with the toxicity profile is actually a little bit different, which is really interesting that all three of these ADCs, even though they're all targeting TROP2 and all have topo 1 payloads, you're not seeing exactly the same toxicity profile, which is, not something I completely understand. And I think it's nice to see these agents in development, but for this patient, the patient with metastatic hormone receptor positive disease, we have data from TROPiCS-02, which led to its approval in this setting. And this trial was specifically for patients who had 2 to 4 prior lines of chemotherapy for metastatic hormone receptor positive disease. They all had to have had a prior CDK, a prior taxane, and then again 2 lines of chemo in the metastatic setting. So it was very heavily pretreated. And again, remember, our patient only had 1 line of chemo, so she actually would not have been someone, for example, who was eligible for TROPiCS-02, given she wasn't quite as pretreated as this population. And so what we saw was that the sacituzumab did significantly better than standard chemo, both in terms of progression-free survival as well as in terms of overall survival. You see the absolute delta in PFS it's about 1.5 months, so I'd say fairly modest. But you see the survival difference now is a little over 3 months, and so I think very clinically meaningful difference there, which I think is important to see. We do also know that even when you look at quality of life, it did seem to extend time to deterioration for global health status relative to chemotherapy. And in fact, it didn't really matter what the degree of TROP2 expression was, that the sacituzumab always did better than standard chemo, whether you were TROP2 high or TROP2 low. I think this is an area of debate, though, that even though, again, we don't need to test for TROP2 because no matter what your result is, still sacituzumab is going to do better than chemo. I do wonder as we get more and more ADCs available if we may need to start thinking about testing, because there does seem to be this correlation that your PFS does seem to get a little longer as TROP2 expression increases with the sacituzumab. And so I think, again, more to come there. But for now, no need to test for TROP2. But I think we do need to be aware that these ADCs do have chemo level toxicities. And in fact, with sacituzumab, half the patients do develop grade 3-4 neutropenia. And, a little under 10% have high-grade GI toxicity. And so a big question that often comes up in clinic is, do you need to give prophylactic growth factor? In fact, 50% of people on the registration trials with sacituzumab ended up using growth factor. And so I'm a little bit cautious about this, and I tend to not use prophylactic growth factor in every patient, but rather use the growth factor up front in someone who's maybe got low blood counts to begin with, or has had prior need for growth factor with prior chemo, and then introduce it only in those patients if they develop neutropenia. And so that has been my general approach. So I think overall, again, the things to think about with sacituzumab are the neutropenia and the diarrhea, and to think about growth factor support. One study that our group did present at ASCO this year was the trial that this patient actually enrolled on, so the SASI-IO HR positive study. And so this was a trial for patients who could have had up to one line of chemotherapy for metastatic disease. So much earlier treatment than TROPiCS-02 because it's 0 to 1 prior lines of chemo. And then they were randomized to get sacituzumab with or without pembrolizumab, but they weren't selected for PD-L1 or TMB or anything. It was just unselected metastatic ER-positive disease. Transparently, we did initially design this trial to select for PD-L1, but it turned out to be very challenging because we found that when we used a CPS-10 cutoff, the prevalence of CPS-10 positive patients in ER-positive disease was only around 6%. And so it just seemed too hard to isolate patients who are CPS-10. We then tried CPS-1, and we still were just finding we were screening too many patients. So that's why we broadened the eligibility and just left it to all comers. In hindsight, maybe that was a mistake, as you'll see from the results. But what you saw was that there was no benefit to adding pembro to sacituzumab. Numerically, maybe the PFS is a little bit better with the two agents, but again, no significant benefit from the addition of pembro. Also, no difference in overall survival. But what was really interesting was that when you did look at outcome by PD-L1 status, in this case, we used a CPS of 1 cutoff just because of the prevalence of CPS-10 being so low, you now do see a pretty nice trend for the sacituzumab pembrolizumab combo doing better. So you see a PFS of 11 months for the combination compared to just 7 months with the sacituzumab alone. And in fact, similarly for overall survival, you see this 6-month difference in overall survival for patients who have a CPS of 1 or greater. And again, these are, small numbers. This is not definitive by any means, but does make you wonder if we had had a well-powered study in a PD-L1 positive population, if that would have been a positive trial. And so there is now a very large study called TroFuse-10. This is a registration trial using sacituzumab deruxtecan, also known as MK-2870, that TROP2-directed ADC, for metastatic hormone receptor positive breast cancer for patients who've not had any prior chemo. So this is their first line of chemotherapy after progressing on endocrine treatment. And it has 3 arms. So they get the ADC alone, or the ADC with pembro, or chemo of physician's choice. And the study's enrolling 1,200 patients. So it will be a huge trial. And so they will have the ability to also look at subset analyses to explore this idea of whether PD-L1 positivity predicts benefit with the combination or if it's just an all-comer benefit. And so I think, again, we need more data here to really understand this. So while, again, that trial with MK2870 is currently enrolling, there's also ASCENT-07, which is trying to move sacituzumab up earlier. Right now, again, based on TROPiCS-02, it was for people that had 2 to 4 prior lines of chemo. ASCENT-07 is for first-line chemotherapy, comparing sacituzumab to chemo physician's choice. And so this study is currently enrolling. So I think the way I think of the current algorithm for treatment is I usually will think about an ADC after someone's had one line of chemo. At this point in time, although I will say this is evolving as we just saw data from Destiny Breast 06, which does position now TDXD as a first line chemo choice, and then would leave sacituzumab, I think, to be really a second line chemotherapy choice in the metastatic hormone receptor positive space. So this is just a nice kind of overview slide of the data that we have across all the ADCs and metastatic breast cancer. I think really just showing the data side by side with TROPiCS-02 with sacituzumab, but then also, putting it in light with the fact that we do have TDXD also available, which, for this particular patient, as we discussed, does have HER2 low disease, and TDXD would be an excellent choice for her as well. I think the challenge with her, though, is that now she's had a topo-1 payload, and so we don't know how TDXD will perform post-sacituzumab, but definitely something I would love to offer her in the future. So we could move on to our next case. This is a 51-year-old woman who had initially presented with a breast mass with an enlarged ipsilateral axillary node that came back as triple negative, but HER2 low. Unfortunately, she was found to have de novo metastatic disease. So at the time of presentation, staging scans did reveal that she had pulmonary nodules that were seen, the largest of which was 2.5 centimeters. So we biopsied one of the lung nodules, which was consistent with her original breast primary, triple negative and HER2 low. We sent off PD-L1 testing, and that came back PD-L1 negative, so CPS was only 1. We also obtained genomic testing, which suggested no targetable alterations. Germline testing was also done, and she did not have a BRCA mutation or PALB2 mutation. So this lady had de novo triple negative disease, just with a couple pulmonary nodules, so very minimal amount of metastatic disease. And so she went on to our trial, actually the sister study for the other trial. This is SASI-IO triple negative, and she got randomized to get sacituzumab with pembrolizumab, and in fact had a complete response. And so remember, she's PD-L1 negative. And so, these trials after you hit, so we stopped the sacituzumab when she hit a complete response, and I left her on the pembrolizumab. But once you hit 35 cycles of pembrolizumab, the trials have you discontinue the immunotherapy. And so we stopped the pembrolizumab, and now it's been a year, and she's been off of everything, and she has nothing and no cancer that we can see on scan. So it's really, amazing to see. Really nice. She's just comes in for checkups now. But I think it does beg this question about, how do ADCs work with immunotherapy, and does it have a role to be given together? And we have some data combining Dato-DXd, so the TROP2 ADC with the deruxtecan payload, given with durvalumab in the Begonia study, where in fact we saw that this combination in the first line triple negative setting was associated with an 80% response rate. And you can see in the bars that patients who are both PD-L1 negative and positive had benefit. You don't know if they would have done just as well with Dato alone. Obviously, this was not a randomized trial, but makes you wonder if the PD-L1 negative patients are potentially deriving benefit even from the addition of checkpoint. And so that's why this trial is ongoing, is it's trying to address this question in a randomized manner of an ADC with or without a checkpoint inhibitor in this PD-L1 negative setting. But there's also an interest in trying to address this in the PD-L1 positive setting, where there are ongoing registration trials as well. We know, for example, that sacituzumab works very well in metastatic triple negative disease. These data come from the ASCENT trial, where sacituzumab was compared to standard chemo in pretreated triple negative disease. And we saw that the sacituzumab did much better than standard chemo. It, in fact, tripled progression-free survival, doubled overall survival. But I think the lesson I've learned here is not just that sacituzumab is a great drug for triple-negative disease, but in fact, when you look at chemo performance in pretreated triple-negative disease, it doesn't do very well. The PFS is only 2 months. It means that most patients are progressing on their very first restaging scan. So really an area where we need to do better. And when you look specifically at the patients in the ASCENT trial who got treated in the second line setting, you see that they do very similarly to the overall trial population. And so we do think about sacituzumab as a standard treatment for second line triple negative breast cancer. But obviously, given the very robust results, we're very interested to see if we can also move it up earlier. And, I think the data we're seeing here is very similar to what we saw in TROPiCS-02, that TROP2 expression also did not predict benefit. But you can again see that PFS does get longer with sacituzumab as TROP2 expression increases. And the toxicity profile is again very much the same as what we know with the neutropenia and diarrhea. But our hope is that when you use a topo-1 payload with an ADC, you can get enough DNA damage that can augment the STING pathway and cause T-cell infiltration that could then allow synergy. And in fact, this seems to be a reasonable hypothesis because we saw some data at ESMO Breast this year combining sacituzumab with atezolizumab for first-line metastatic triple-negative breast cancer that was PD-L1 positive. And you saw almost an 80% objective response rate and a PFS that was 12 months, so really much longer than we would see with chemo here. And so I think, again, we're very optimistic for these registration trials that are combining ADC with checkpoint. There's ASCENT-04, which is nearly complete with accrual, that randomized patients to sacituzumab with pembro or to chemo pembro. And then TROPION-Breast05 is enrolling with Dato-DXd with or without durvalumab compared to chemo pembro. So, I think, again, these studies, given what we've seen, I think are very promising. And hopefully we will also see sacituzumab move up into the first line PD-L1 negative setting because we also have trials ongoing there with ASCENT-03 and also TROPION-Breast02. But, we talked about this new ADC on the block, if you will, the SAC-TMT, this TROP2 ADC also with the topo-1 payload. And at ASCO, we actually saw data for this drug in a phase 3 trial that was conducted in China. It is almost identical to the trial design for ASCENT. So it really took people who were in the third line or beyond metastatic triple negative setting and compared SAC-TMT to chemo of choice. And the data, again, looks almost identical with the progression-free survival being about tripled and overall survival also being about doubled. So I think, again, looks like a very promising ADC, and you can see benefit was seen irrespective of TROP2 expression. But what was really fascinating to me is that the toxicities are a little different. So you do see neutropenia occurs just like with sacituzumab, but it's a little less common. But you also see stomatitis. And so we don't usually see stomatitis with sacituzumab, but we do see it with Dato-DXd. And so it kind of seems to me like it's got toxicity profiles a little bit in between Dato-DXd and sacituzumab, which, again, is really fascinating how these ADC tox profiles are a bit different despite being so similar mechanistically. So we'll have to see what happens. There are now multiple registration trials about to start with SAC-TMT as well. But for now, I think sacituzumab remains a second-line standard of care. But I think we're likely to see data from the trials looking at sacituzumab as well as Dato-DXd in the first-line setting, probably in early 2025. So I think just finally, the last case is this patient who had de novo metastatic ER-positive breast cancer that was also HER2 low. She had been treated with an AI and CDK up front and had a great response and was on it for over 30 months and progressed and did not have a PI3 kinase or ESR1 mutation. So I treated her with fulvestrant everolimus, which she did okay on for about 7 months. And then went on to capecitabine and had a wonderful response, almost 2 years on capecitabine, and then enrolled on TROPION-Breast01. And so this is the trial that compared Dato-DXd to chemo physician's choice, and she was randomized to Dato-DXd. And she did have a partial response to Dato-DXd and did great on it. But afterwards, I put her on TDXD directly after progressing on Dato-DXd, and she progressed on the TDXD on the very first scan that she had, and then has been subsequently on paclitaxel. But so I think it brings up this question: is what's the efficacy of Dato-DXd in this setting? Again, this is another TROP2-directed ADC that has in this case, the deruxtecan payload, so the same payload as trastuzumab deruxtecan does, but the drug-to-antibody ratio here is lower. So it's a drug-to-antibody ratio of 4-1 as opposed to sacituzumab and the sac-TMT agent, which both are about 8-1. And when you compare Dato-DXd to standard chemo, this was done in the TROPION-Breast01 trial for patients who had had 1 or 2 prior lines of chemotherapy in the metastatic setting. And you saw that Dato-DXd was associated with about a 2-month improvement in progression-free survival. So it did improve PFS. The overall survival data is still immature. And we've also now seen data for PFS2 also suggesting it is prolonged in the Dato-DXd arm. The response rate is higher with Dato-DXd compared to standard chemotherapy. But what's interesting, again, is the differences in the toxicity profiles, because what you're seeing with Dato-DXd is, in fact, there are a lot fewer grade 3 toxicities with Dato compared to standard chemo. This is kind of unusual because most ADCs are not noting less in the way of high-grade toxicity, but this agent is. And I think that's because it does not cause neutropenia. So you're not seeing all the high-grade neutropenia like you see with sacituzumab. But what you do see is stomatitis. And so you do have to keep that in mind, that about half of patients will develop stomatitis, most of it being low grade. In this trial, patients were not mandated to get prophylactic treatment with steroid mouthwash, though I will say that I do give everyone prophylactic dexamethasone mouth rinse if I'm going to give them Dato-DXd, and I think it works quite well. You can see dry eyes as well with Dato-DXd, so important to mention to patients to use rewetting drops. And then ILD is super rare with this. So it's only about 3% of patients that experience ILD, very different than TDXd, which, we usually see a 10 to 15% rate of ILD. So that's reassuring. But I think the issue we're going to run into is, we're waiting on approval for Dato-DXd. And let's say we do get an FDA approval. The question is going to be, how do we actually integrate this into our practice? Because, Destiny Breast 06 just taught us that TDXd works really well in metastatic hormone receptor positive disease as the first chemo choice. So that kind of leaves the question, well, will a TROP2 ADC work after TDXd, especially if we're delivering topo-1 payloads sequentially? So I think we need more data here, and there are now lots of planned trials to address this. This is one such study being led by my colleague Ana Garrido Castro, where we're giving sequential TDXd and Dato-DXd one after the other. And so this will help us understand kind of how these drugs work sequentially, and it will have very rich biopsies done to help us understand resistance mechanisms here. So hopefully we'll learn more. But for now, I think most of us are sequencing these ADCs, even though we don't have great data for that.